Hello, my name is Derek Nola. I'm a junior studying computer engineering. My topic today is about the simulation of biological cells, specifically the current state of computer simulations of biological systems. This is directly related to my major and the industry of large-scale computing. The format for this presentation is Pekka Kucha. This was invented by Astrid Klein and Mark Ditham in 2003. They claim they are tired of listening to architects drone on and on about their projects. The format is a presentation composed of 20 images, each 20 seconds. I have modified the style somewhat, allowing for multiple pictures per slide and reducing the number of slides to 19. This is to reduce the length of the presentation to better fit with time constraints. This format is similar to Ignite talks held at Cal Poly every quarter. To quickly review, a general biological organization is shown. This presentation will largely focus on the three areas highlighted: cells, tissues, and organ simulations. It will also cover simple organism simulations. It is important to look at traditional cell studies first. While it is easy to duplicate cells in a laboratory, these studies are expensive. Traditional analysis can be quite complex, with many factors working in parallel, forcing studies to hone in on individual aspects of a cell. It should also be noted that a large portion of biological models are recursive in nature, so computation is needed. Computer simulations arise from taking a real-world problem, abstracting it to a single or series of mathematical functions, and then creating software that computes these functions continually over some period of time. Conclusions can then be drawn from these simulations and real-world knowledge gained. What results is a simulation that shows how hurricanes behave or the possible bendings of an alpha helical protein. Looking at cell simulations in particular, several software packages are commonly used. The most common is eCell, created in 1996, which is a Python-based software package for defining cell and other biological models. It also includes a graphical user interface to create causal relations between cellular components. Since cellular simulations are relatively new, many projects create their own proprietary simulation frameworks. The first level of cell simulation is modeling individual organelles. An example is mitochondrial modeling. This is particularly important as several diseases exist because of mitochondrial failure. This study was complete in 2008 at Purdue University. It sought to model the creation of ATP and other energy-producing molecules while integrating the volume dynamics of mitochondria. This simulation uses 359 parameters to generate results. If this sounds like a lot, the scientists used MATLAB and Monte Carlo algorithms to reduce the number of independent parameters from the original models. They also use local sensitivity control algorithms to reduce the error in the model. This was done on a cluster of 4 Intel Xenon processors, each with 16 gigs of RAM. Looking at the results, the simulation model is shown in the solid and dotted lines, while the real-world experiments are shown in the symbols. Simulating the mitochondria under a variety of environments, you can see that the simulation closely matches the real-world results of an actual mitochondria. Going forward, scientists can use these models to simulate new drugs and treatment options for mitochondrial diseases. The next level of cellular simulation is whole cell or multi-cell simulations. In a landslide Stanford research study in 2012, scientists were able to completely simulate Mycoplasma gelatinium, the world's smallest free-living bacterium, with only 525 genes. Since its release, this article has been cited 693 times by other studies. The simulation used over 1,900 parameters. These were divided into 28 modules, each with its own algorithm. One pass took 128 wild-type cells and then simulated them until they had all replicated. A single pass took almost 10 hours with MATLAB software running on a 128-core Linux cluster. The study generated an incredible amount of data. Each pass of the simulation produced around half a terabyte of data. The simulation was so extensive that the model was able to predict the phenotype of the bacteria just from its genotype. Shown here is an 8-minute run of the simulation, sped up to 20 seconds. One thing to note is the real-time tracing of the cell replication on the right. Next up is modeling simple organisms. Started in 2013, the Open Worm project is attempting to completely simulate the transparent roundworm C. elegans. C. elegans has both its nervous structure and genome fully mapped. It is one of the simplest nervous systems of any organism, with only 302 neurons and just under 1,000 cells in its entire body, each with a unique identifier and supporting literature. Currently, the project is attempting to model the worm's locomotion by simulating only 302 neurons and 95 muscle cells. While still in development, significant tools have already been produced, such as the neuroconstruct, which is able to output neural structures in neural ML. This is used to reconstruct the full neural connectome of the worm as well as build the model muscle cells. The virtual worm first takes in environmental stimulus. External cells react, sending information through its neuron network. In response, motor neurons fire, which causes the worm to move, changing the environment and beginning the cycle once again. The neural cells are being modeled by Hodgkin's-Huxley equations, while motor cells are being modeled by a smooth particle hydrodynamic algorithm. This is a prototype simulation. The worm is in shallow water and struggles to move. This video is actually a quarter second in real time, but took 47 hours to render on a quad-core Xenon processor. This project is entirely open source. Anyone can work on it. All scholarly articles are published and available for free. Once finished, larger organisms will be simulated using the tools created by this project. Beyond these projects, greater simulations are possible. Biological simulations are compute hungry and will remain a forefront of computation. Even as Moore's law of increasing single-threaded performance, shown in blue, comes to an end after 60 years, the rapid growth in cluster and parallel computing, shown in green, promises a future of constantly increasing computational power. This increasing power, combined with new parallel algorithms and the knowledge learned from current simulations, will enable larger cellular simulations. Simulations like the Blue Brain project, shown here with a brain of only several thousand neurons firing, will likely reach their goal of simulating an entire human brain within the next 50 years. Ultimately, the simulations today are going to greatly benefit the study of biology. Computational models can simulate complex biological systems rapidly, allowing a large variety of drugs and molecules to be tested on cells. Cells' responses to potential new medicines can be virtually tested before any type of physical trial. In the end, a greater understanding of our world and the new forms of medicine are possible with advances in computer modeling. Thank you for your time and have a good day.